Platinum is such a ubiquitous agent, and our lead program is in ovarian cancer. When it works extraordinarily well. Unfortunately, it stops working for far too many patients. And gynecologic oncologists have long referred to the potential to resensitize tumors to platinum as their holy grail.
Welcome to Power to the Patients. I'm your host, Brandon Lee, and I'm joined today by Tom Zindrick, who is a biotech exec, a life science strategist, and the CEO and president of Genelux Corp. So today we're going to talk about some of the really exciting work that you're doing in oncolytic virus therapies. But before we get there, Tom, maybe just give me a quick kind of thumbnail sketch of your career and how you got so excited about the work that you're doing today.
Great. It's a pleasure, Brandon. Really appreciate the opportunity to be here today and talk to you on your platform and share a little bit about our story. My backstory was not a direct line from kind of where I started to where I am today. This is typical. I actually have an undergrad degree in biology. Then I went to law school, started work as a patent attorney. And while practicing as an attorney, I went back and pursued graduate studies in molecular biology. So I would practice all during the day, and then I would go into the lab at night and work on gels back in the day, making single-chain bispecific antibodies. So really kind of a varied background, but it was all directed to ultimately where I landed, where I spent the bulk of my career which is at Amgen. I joined Amgen in the early 90s in the legal department, starting out as a patent attorney, but also assumed business development responsibilities. I was company sole transactions attorney starting in the mid-90s and built up that group over a decade. I also took on responsibility for the FDA regulatory and compliance group within the law department. And so had an opportunity to sit on a number of product development teams that went to market, working with some world-class drug developers. Also had the opportunity to work on almost every transaction the company did through logarithmic growth. So it really gave me a broad exposure and aperture to biotech development and the business of biotech. And then my last significant role at Amgen, starting in 2004, the board of directors approached me and asked me to report in directly to them as the company's first full-time chief compliance officer. A company had grown so rapidly and really had outgrown its systems, and compliance was becoming much more significant. And so they asked me to help organize that organization and really develop it into what it is today. So I'm very proud of the work that we did at Amgen. When I joined, we probably had 2,000 employees. When I left, we had 20,000, to give you an idea of the growth trajectory that we were on and the opportunities that was really afforded me and others that were part of the company.
Wow. I think one of the things that kind of stands out about that is the transition from leading compliance to being the CEO of a biotech. Tell me about that kind of transition and how that happened.
Yeah. Again, it really comes down to relationships, trying to do your best every day, and then not really sure where it's going to take you, but usually good things happen. And in this case, after I had left Amgen, I received a call from some former executive leadership team members who were looking at this company in San Diego that I had never heard of, Genelux, and they were thinking about joining the board, and asked me to go down and really kick the tires on the company for them, get a better understanding of the science, the team, and really the development program. And I did that for about 6 months, and it was a great exposure to the company. I consulted. I was offered the opportunity to become chief operating officer during that period. But the company was headquartered in San Diego. I was still living and continue to live north of LA in Thousand Oaks, where Amgen is located. And I had a younger family at the time, so I couldn't make that transition. But it did give me the opportunity to not only meet the staff and get to learn a little bit more about the science, which I found absolutely fascinating. But I also was invited to host the 2010 shareholder meeting, where I met some key shareholders. And it was at that point, or from that engagement, that in 2014-15, the board contacted me, asked me if I would be willing to come back as CEO. The company had been, up to that point, run by the founder scientist who was really focused on basic research, and very proud of the work that the team did and understanding the fundamental biology. What the board was really looking for at that point, as is typical in the evolution of any company, biotech or tech or otherwise, was that transition from basic research into translational research in medicine with a go-to-market strategy ultimately. And I felt with my background, I could be of some service, and it's just been a phenomenal ride. I've got a fantastic team here, really dedicated individuals who are committed to the platform and the patients. And that's what drives us every day.
Yeah, that's a wonderful tee up. I'd love to maybe just start with the big idea. What is one big idea that you want listeners to really understand about the work that you're doing at Genelux and the role of oncolytic viruses?
Absolutely. So oncolytic viruses are in the broader category of immunotherapy, which is, I think, the most exciting development in oncology today. It's, in certain indications, it's really helped to bend the survival curves, but it's also been somewhat frustrating because for far too many tumor types, it may otherwise appear that the patient's immune system should react and respond and attack a cancer. That doesn't happen as aggressively as we'd like. And so, with what we're doing in the oncolytic virus space, and specifically with our lead agent, Olvi-Vec, is that I think of it as like an off-the-shelf, personalized medicine in the sense that we self-manufacture Olvi-Vec. It's the same presentation for every patient. But because of the unique aspects and characteristics of an oncolytic virus, we're able to prime and target the patient's immune system to be able to attack the patient's own tumor. Because with tumor heterogeneity, every tumor is unique to every patient. And being able to really tailor the immune attack to that patient's own tumor while using a convenient agent that doesn't require excessive engineering and expensive production processes is, we think, primarily when we think about the oncolytic virus modality, it does things in a unique way that I don't think any other agent has combined. It directly kills tumor cells. As I mentioned, it then stimulates an immune response against the tumor. And all of that creates a local inflammation that remodels the tumor microenvironment. And with that, that conversion of the so-called immunologically cold tumor types to the immunologically hot not only causes that direct and indirect immunotherapeutic attack on the tumor, but what we have found and what we're really pursuing clinically is that during the course of remodeling, we can also resensitize tumors to frontline therapies. In our most advanced program, which we'll go into, is initially focused on resensitizing tumors to platinum. Platinum is such a ubiquitous agent, and our lead program is in ovarian cancer. When it works extraordinarily well. Unfortunately, it stops working for far too many patients. And gynecologic oncologists have long referred to resensitized tumors to platinum as their holy grail. And we get through a really, I think, extraordinary set of results in phase 2, have positioned ourselves in an ongoing phase 3 to definitively prove that up, that we can resensitize tumors. And we're pursuing that not only in ovarian cancer, but in a couple of other trials as well.
So much to dig into here. And I want to start at the very top, which is in the grand scheme of cancer treatment. How do you think about immunotherapy versus, let's call it, like traditional cancer treatments? And then within that, I'm curious, what is it about an oncolytic virus that is specifically different from, let's call it, the rest of immunotherapy as a category?
Yeah, I see them all ultimately working in a complementary fashion. So while we can segment them by categories, mechanisms of action, because of the unique characteristics of Olvi-Vec that I've laid out, when I look at other therapeutic agents, my first reaction isn't to think of them as potential competitors, for example. I view them as potential collaborators because of the variety of modes that we can then bring to bear on the tumor, the anti-tumor activity. It really sets us up to be able to combine. I think what was apparent to me when I joined Genelux is based on early preclinical models, there was a perception, and these are often nude mice with incomplete immune systems. There was often a perception that oncolytic viruses could carry the whole load. But as we know, it's quite a complex step to go from animal models into humans. And as we've learned in working with immunocompetent animals, there's that immunity paradox, right? The body is evolved over the millennia to attack and remove viruses and bacteria and the like. So it's drawing that balance of being able to deliver an oncolytic virus that's been genetically engineered to be specific to being an anti-cancer agent and to be able to stimulate the immune system while still understanding that the body's constantly trying to stay in a state of homeostasis. And so we've been working in that regard to thread that needle. So I see within the immunotherapy perspective, if you think about the three components, direct cell killing, you can think of maybe a substitute being engineered bacteria, for example, or immune priming. You can think of maybe sting agonists as potential. With respect to the remodeling of the tumor microenvironment and platinum resensitization, I really don't know of a good substitute for that. So I think that the really exciting aspect is that while there may be individual components that generally could be a proxy for other agents, none really encapsulates it in the same powerful way that I think the oncolytic virus modality has the potential to do. In terms of targeted therapies and the like, clearly there's been huge progress made in that regard, and those have been really transformational. However, as we know that too often with a targeted therapy, whether it's through escape mutations or some other feedback loop, what can initially be a very pronounced effect can often then turn into an aggressive relapse as the tumor evolves and or adapts. And in this case, for us, we believe because of the ability to prime the immune system and combine with other agents, we're not limited to or have a risk of a mutation allowing the tumor to escape. We're coming at it from multiple angles to try to overwhelm the tumor's defenses.
One of the things that you mentioned that I'd love to understand a little bit more about is the personalized nature and the requisite manufacturing. Can you help unpack that a little bit? Like, what does that look like?
Absolutely. So in our case, we self-manufacture Olvi-Vec at a facility we control in San Diego. It's a closed batch system bioreactor, and we produce the same version of Olvi-Vec with every batch. And then we ship it cold chain storage, and then ultimately it is administered to the patient in the clinic. In the case of ovarian cancer, we administer it through a catheter intraperitoneal route administration, not an uncommon way for chemotherapies to be administered by gynecologic oncologists. In some other studies we have, two other registration path studies we have, we're administering it systemically. So we do have ultimate flexibility in how we deliver Olvi-Vec. The personalized component of this is, so just to kind of unpack a little bit about what happens when the virus infects or is administered. As I mentioned, you got to overcome the delivery issues, whether it's the immunity paradox or it's physical barriers, the extracellular matrix, the hostile environments, whether it's hypoxia and the like. But once you do that, well, in our case, vaccinia, unlike a number of other viruses, it's a large double-stranded DNA virus that infects a tumor cell through the membrane. So it doesn't require a biomarker or a receptor. So it allows for a broad spectrum of activity. We've genetically modified Olvi-Vec so that it is attenuated, meaning that we control its replication cycle. So we can, from a safety perspective, it's that good balance. Vaccinia is a cytoplasmic virus. And so it effectively brings its own host cell machinery to do its replication. By knocking out a key gene, a thymidine kinase gene, as we have done, it then makes the virus dependent upon an abandoned pool of nucleotides. While this modified virus might get into healthy tissue, A, it doesn't have the substrate to replicate, and B, while it's sitting there in a healthy immunocompetent tissue, it gets cleared very rapidly. On the other hand, in a tumor cell where the virus does have the nutrients to replicate itself, it's really, think of it as a Trojan horse that cancer cells have disabled the antiviral components to enable the tumor to be cancerous, whether it's p53 or some other host cell components. And as a consequence, the virus takes over the host cell machinery for its own purpose and replicates. And at a certain point, it causes that host cell to burst. It releases not only additional virions, the neoantigens to the immune system, along with the DAMPs in a way that triggers the immune system, and through antigen cross-presentation, sends these antigens to the immune system so that as a tumor is constantly evolving, those unique neoantigens are presented to the patient's own immune system. And we demonstrated in both preclinical and clinical translational studies that show that we can generate an anti-tumor T cell response in the blood. We demonstrate that CD8 positive T cells, effector cells, infiltrate into the tumor epithelium. And then with that immune cascade that comes down, what we've learned, and this comes back to really starting to appreciate a virus or optimizing and letting the virus do what it does best, right? Rather than try to put a lot of different bells and whistles on the virus and hope that it alone can eradicate a tumor before the immune system clears it. We really think of it more as a priming for a catalyst or a catalytic event. So we don't have to infect every tumor cell. We just have to infect and kill enough tumor cells to activate the immune system. And there comes a point where it's almost a tipping point that we've seen preclinically, that when we're able to do that, you see a profound impact on anti-tumor activity. And again, what's unique about working with an oncolytic virus is we don't think of administration in a typical PK load perspective because it's a live agent. And there we know that the virus has taken hold, it's formed the beachhead, and it's having a profound replicative effect that we ultimately recognize will then stimulate the immune system and again, modify certain biological pathways. Maybe if I can't, just to finish up on that point, the line of clinical data and thinking that we're trying to pursue is we noted from some of our RNA analyses that several of the biological pathways, one in particular in STAT1, has been implicated in platinum resensitization. Tumor fibroblasts, the glutathione pathway can downregulate, which enables the tumor to expel platinum. We showed the ability to upregulate STAT1 in a statistically different way. And we believe that the effector cells, whether those are against the virus backbone, some of the payload proteins, or of course the tumor neoantigens, any of those we believe has the ability to have that reversal effect on the STAT1 pathway. And as a consequence, what occurs is you get initial viral infection, the immune response, both the direct and the immune cell death, but you also, through that, have set the tumor microenvironment up to be re-challenged by platinum. And when re-challenging by platinum, and when the platinum isn't removed from the tumor, it does two things. It obviously kills tumor cells. It also abrogates a number of the suppressor cells. And with that, it then will resensitize the memory T cells that were generated from the original Ovvi-Vec infection. Probably the most profound example I have of that is in our phase 2 study. We had a platinum-refractory patient, heavily pretreated. And by definition, a refractory patient is a patient who, on her last exposure to platinum, progressed while on drug. So the tumor was absolutely recalcitrant to being treated. This patient, after a single cycle of Olvi-Vec, so patients received doses on 2 consecutive days, the catheters removed, that patient went from no response to responding for 17 cycles of platinum, which is kind of in its own right a case study in terms of being able to tolerate the platinum, and her quality of life, as reported to us, was quite high.
The way this works, I'm going to explain it as if I have a 5th grade understanding of it because that's about as much as I have. So it sounds like you've developed a virus that specifically replicates in tumor cells and not in healthy cells. When it replicates, the cells burst. So this is kind of like a tumor killing effect. And then in the bursting of the cell, you then trigger an immune response for the body to then go further target tumor cells of that kind. I guess like the piece that I haven't quite caught is like, what is it about that resensitizes the tumor to platinum?
So that's a great question. So there's a really elegant paper that was published in Cell by a group out of the University of Michigan. And what they demonstrated is in tumor cells that had become platinum resistant, they showed that through tumor fibroblasts and interferon gamma pathway and other chemo and cytokine releases, there would be modification of the STAT1 pathway through glutathione. And they were able to reverse that platinum resistance. And so you're exactly right. Through the immune system activation, as I mentioned, shown that there are effector T cells that infiltrate into the tumor epithelium. We believe, based on connecting a lot of these translational dots, that those affected T cells work exactly the way was shown in the cell paper. They approach the stroma of the tumor, and are able to overcome the fibroblast-initiated platinum resistance, therefore resensitizing the tumor to platinum, putting it back in its original state, and therefore making the tumor susceptible to rechallenge. Not only do you get that typical platinum cytotoxic death, but because of the immune response that you have there, you effectively created a so-called hot tumor. And that's why we believe you get this so-called prime and boost.
Fascinating. How should I be thinking about the lines of therapy in a world where you validated, like in the phase 3, that this works as kind of hypothesized? Because it sounds like it creates a virtuous cycle with platinum. Help me understand that.
In terms of the lines of therapy, so in the case of, and this is something that Richard Pazder from FDA just wrote an editorial that was in JAMA, where he's pointed out that there now needs to be an appropriate appreciation for the heterogeneity that is caused through multiple lines of chemotherapy or other therapies. Because as we know in our knowledge and thinking about the tumors have evolved, every time there's an intervention, the tumor adapts. In our case, what's really exciting, as I mentioned earlier, we don't need a biomarker for Olvi-Vec to infect and replicate and kill. We had patients that had between 2 and 9 prior lines with a median of 4. And what's really exciting to us is as we look at the patient demographics, patient treatment history, the genomic markers of that patient, and the different lines of therapy or platinum classification, whether it was resistant or refractory, there is no delta in response across any metric that we measured. Which what we get excited about is while I talk about other therapies potentially being collaborators, so that's where the targeted therapy drive has taken us, is the segmentation of patient populations. In 7 completed trials prior to the 3 that we have ongoing, we treated over 150 patients, different tumor types, different demographics, different routes of administration. We've never hit an MTD, never. So we believe we are delivering an immunotherapy that at least rivals, if not exceeds, the safety and tolerability of many other newer agents, but has that broad spectrum of activity of a general cytotoxic. So we believe we can bring to bear an immunotherapy that has many of the advantages of the older therapies with the general cytotoxics. And while we are not limited to combining with platinum, I believe we're only starting to scratch the surface. So in one of our other trials in non-small cell lung cancer, where we are treating a recurrent population that failed frontline, and that is platinum and checkpoint inhibitor, and we're re-challenging with both platinum and a checkpoint inhibitor because we've shown that we can increase the expression of PD-L1 in patients who would otherwise be considered negative. So we think there's an opportunity to catalyze with a variety of other agents. And effectively, our goal would be to become a kind of a backbone for a number of different combination of therapies.
I guess like the natural kind of like follow-up to that is, how should we be thinking about tumor types that you are bullish on this being productive for and tumor types that you're not? It sounds like, how should we be thinking about that?
It's a really good question. So part of it starts with delivery, which are most accessible. There are other later stage oncolytic virus programs that are showing great results in skin cancer or in bladder cancer, which tend to be through direct administration. We started in the ovarian space through this intraperitoneal or catheter delivery because we felt like that would give us an opportunity with a new dosing regimen that we were testing, to give us the best opportunity to demonstrate anti-tumor activity. We were working in a smaller volume, and ovarian cancer forms a sheath on the abdomen, so a lot of surface area to put virus on tumor. So we really thought we gave ourselves the best opportunity to kind of stack the deck, and certainly has borne out. That having been said, in several of those 7 completed studies that I've pointed to, we demonstrated the ability to administer the virus IV or systemically. And in one study in particular, in patients with a variety of solid tumors, for those patients in particular who had lung disease, whether it was primary or metastatic, we showed a dose-dependent overall survival benefit as a monotherapy in patients that had a median of 5 prior lines, with the first pass effect and the high vascularization of the pulmonary circulatory system. We think that gives us, again, a great entry point into systemic administration. We don't think we're limited there, but we believe that's a great place to start. And whether it's ultimately moving from lung cancer to want to get really creative, think about any patient with a lung met of a solid tumor of any origin, where we can create that beachhead in the lung organ to generate that whole body immune response and go from there. But the virus gets through the entire body. So again, we're not limited, but as we move forward, we're kind of thinking about this in a stepwise fashion because we haven't failed a clinical trial yet. I don't want to be the one that does. We're certainly positioning ourselves to be thoughtful. I think ultimately, maybe to come back to your question, which I didn't mean to avoid because it's such a target-rich environment, if you will. If we're going to pursue, and I think the best approach currently is to pursue a combination approach, then to me, the next frontier is how do we mix and match combination therapies? And that may then dictate where we go from a tumor type. And the reason I say that is in the ovarian study, prior to our moving into the phase 2 with a combination, we were doing a phase 1b dose escalation study as a monotherapy. And we were able to get paired biopsies pre and post administration of Olvi-Vec. And it was there that we analyzed, as I mentioned, the generation of anti-tumor T cell responses, the multiplex IHC, the infiltration of these activated T cells. And when you line those up, what was encouraging was that Olvi-Vec almost consistently was doing what we wanted it to do in every patient. But then when you overlay the clinical response, it didn't match, right? You couldn't point to the infiltration of T cells. So you think about the paradigm of turning a cold tumor hot and activating the immune system for the infiltration of T cells. Why didn't it work in some patients versus others, right? And part of that's the tumor microenvironment. Some of it may be that some patients, platinum isn't the right combination. It might be a checkpoint inhibitor. It may be a targeted therapy. It could be whatever. That to me is really the next frontier for us. To me, that only then creating the next order of questions that are so much bigger than even the ones we're pursuing right now, which is if you have a tailored agent, as we do, how do we tailor the therapy with other combinations to be effective for the maximum number of patients that we can?
I'm hearing a lot of things that make this sound perfect, but in any kind of emerging field, like any sort of therapeutic platform has fundamental constraints of science, constraints of physics and biology. What are the kind of constraints or limitations that researchers are like staring at that are kind of known constraints? And then what are the constraints that are the ones that I guess you're kind of like waiting for the data cards to turn over that you're thinking through as potential risks here?
Yeah, I certainly don't want to suggest that this is a panacea. There are many unanswered questions that we're in the process of answering some. One of those is the immunity paradox, right? We want to stimulate the immune system. We want an active immune system, but that same immune system can be called against the virus and remove it, whether it's through complement, the innate immune system, or antibodies in a way that can blunt its effect, right? So it's kind of a race against time. In terms of getting enough virus in to be able to prime the immune system. That's why I like the combinatorial approach. Historically, there have been some concerns because of structural physical barriers. Can you get enough virus into the tumor? And oftentimes you'll see patchy infiltration and replication. Another aspect is the lytic activity. The one approved oncolytic virus on the market today is we use a vaccinia virus. It's a non-human pathogen. It was designed as a vaccine. So its backbone was designed to light up the immune system. That's why we think it's an ideal oncolytic virus. Other therapies like being used off of or developed off of herpes virus backbones or adeno or cold virus backbones, whose backbones have evolved over the millennia to evade the immune system. And oftentimes you need to put on pretty strong payloads or stimulatory payloads, and that can come with its own challenges. So there are concerns about can you, through the lytic activity, do enough to stimulate the immune system? We've demonstrated through intraperitoneal administration, the infiltration, the killing and infiltration of immune cells into the abdomen. We've never found a live virus outside the abdomen, but in patients who had lung mets, malignant pleural effusion, we saw a similar dynamic in the pleural space as we did in the abdominal cavity, where you saw that infiltration of immune systems. So you need to have a sufficiently strong lytic priming agent in order to be able to generate that. And as I mentioned, the original and the first approved oncolytic virus really was effective in the local regional direct injection, but was really never demonstrated a strong activity distally. So that's something that ultimately we're very confident based on our phase 2 data. But we have a phase 3 study that we've guided that will read out by the end of the year. It's a randomized trial, a 2-to-1 randomization. And we'll ultimately wait for the data to prove up and hopefully confirm why we're so excited about where we are currently. Other sort of practical questions come up around handling of the virus. Is it safe? Can it mutate? Again, vaccinia is a large DNA virus. It comes with its own proofreading mechanism. It also is a cytoplasmic virus. And so there's no risk of host cell integration, and it needs a tumor in which to replicate. So we do pay close attention to viral shedding. Is there an opportunity through bodily fluids or through pox-like lesions to transfect, get in the environment and transfect? That's always a theoretical concern. It's one we watch, and we're doing viral shedding as part of the phase 3 study to confirm that that's not an issue. And I think maybe the other concerns are around manufacturing and how cost effective can an oncolytic virus be. Historically, they've been fairly expensive to manufacture. They're also BSL-2, so Biosafety Lab 2 category in the manufacturing setting. So certain protections need to be put into place. In the clinic, it's Biosafety Lab 1, which is the least, considered the safest. But in larger volumes, it's a BSL-2. And so we have to be very cognizant of the manufacturing process, but we have a closed loop batch process where we have great sterility. We can control it, and it's a proprietary process that we've implemented in our facility in San Diego where we can scale in a very cost-efficient way. So we believe we have addressed many of the concerns around manufacturing. And then kind of post-COVID, because we keep the virus at minus 70 currently, cold chain storage and distribution is always a question. But post-COVID, where almost every center now has a minus 70 freezer for the vaccines, it hasn't been an issue for us at all. So there have historically been a number of concerns, and like a lot of modalities can be overlooked for a number of years. We believe we're getting to a point where the companies who have stayed the course are soon to be addressing many of the impediments that were perceived as real headwinds to oncolytic virus therapy, being a significant part of physicians' armamentarium.
The thing that I'm kind of left thinking about here, Tom, is like the second order effects of if this all goes well. In particular, one of the things you've kind of described is the like resensitization, platinum and other potential therapies. And then as a result, what happens to like the kind of status quo of cancer treatment, which is so like sequential in nature today?
The way I've been thinking about it is through our clinical studies, there are really two redefining goals that I have. One is to redefine the oncolytic virus modality through IV administration, something that has long been understood to fully unlock its potential. And the other is to redefine certain treatment paradigms, and platinum resensitization being the first of that. When I look at our phase 2 data, we're dealing with patients for which there is no standard of care. That right now they have physician's choice. Almost every clinical study out there caps the number of prior lines. We start at a minimum of 3 prior lines and go from there. So we're taking patients who, frankly, when they enrolled into our trial, typically considered to have somewhere between 6 months and less than a year of survival. In that case, you're often thinking about so-called salvage therapy, if you will. But in our phase 2 trial, across the platinum-refractory and resistant population, we took a patient population that in their immediate preceding line of therapy had a 4.5-month PFS. Had they not joined our trial, historical data would suggest that receiving physician's choice, they probably could expect a 4-month PFS. Our patients had an 11-month PFS. So we're basically taking an all-comers, most difficult to treat population and giving them a PFS that in some instances, Elahere's PFS for a patient population that could have no more then 3 prior lines, and it was segmented by needing a biomarker, was 5.5 months. We doubled that using platinum chemotherapy in this recurrent setting. So to your point, I think we have this opportunity to kind of redefine what late stage means in that regard. And we're doing it with relatively inexpensive chemotherapies. As I mentioned, we've got patients who tolerated chemo extraordinarily well, in part, and I'll give credit to a number of these newer therapies. I do believe because of the improved therapeutic options for patients who have relapsed frontline platinum, there is a longer time off platinum. Everything we've seen does not suggest that time off platinum means they will do better on platinum subsequently, but it does give the body time to sort of recover from that initial platinum treatment. So they're just in a better performance status to be able to then be re-challenged with platinum. So I do think there's benefit to that sequence, but from my perspective, our goal would be to jump upon approval in this late stage population and go directly into frontline. And in fact, during several conversations we've had with the FDA, that's exactly what they told us to do. And their point was they see everything. And given what they've seen that we can do in these most difficult to treat patients, they're very excited about what we might be able to do in treating patients who are naive to treatment, whose immune systems are intact, and whose tumors have not become hostile to therapy because of all these adaptive processes that it implemented through challenge after challenge of prior lines of therapy. So I think there's an opportunity to effectively transform late stage and potentiate frontline and basically collapse everything in the middle, ultimately. I think in a way that really helps expand. I don't think it's a zero-sum game. I think we're expanding the universe of what we can do for patients.
Well, Tom, that's a powerful vision, and I think an appropriate place for us to kind of wrap today. I want to thank you for taking the time to join me on the show and teach me a little bit more about what you and the team are doing over there at Genelux.
It's my pleasure, Brennan. I look forward to coming back at some point when I've got data from some of these trials and really digging in to see how close we've come to our hypothesis and where we take the program from there.
We'll love that, Tom. Thanks again.
Thank you.